Hi everyone. I'm Phil Garner. I'm the director, one of the directors of the PCSIG, and I'm hosting today's webinar in the absence of Neil Spooner. So welcome to the webinar. Today's webinar is titled Biomarkers of Colorectal Polyps from at-home collected dried blood spots, and presented by Ben Herbert of Sangui Bio. Before handing over to Ben, I just want to take a moment, as usual, to thank our the PCSIG sponsors, which are shown here. Without support of the sponsors, these webinars would not be possible. So thanks ever so much for that. And the last piece of housekeeping before we kick off, Ben will be taking questions at the end of the main presentation, as we usually do on these webinars. So please log your questions as we go by using the chat function that you'll find on your screen. You can do this via public chat, which will be seen by everyone, attendees and speakers, or you can use private chat, which would just come through to Ben and I. Also note that the presentation will be recorded and available on demand after, if you want to share it with colleagues. So without further ado, I'd like to hand over to Ben. All yours, Ben.
Thanks, Phil, and hi to everybody. Hope all of you are not up at 1 o'clock in the morning like I am here in Sydney. But anyway, nice to be here. So I think if I use the down arrow, there we go. So what I thought I'd do first was take you through a bit of the background of Sangui Bio and how we got to the way we use dried blood spots, and basically everything we do, we use Mitra, Neoterix Mitra devices, mostly 30 microliter ones. But originally we worked with venous blood, conventional venous blood, and we were mostly looking at cytokines as markers of disease, particularly inflammation. And what we were seeing was that once that got out of our lab and we were getting blood samples from other labs, or particularly hospitals, that we were seeing signals that indicated that cytokine levels were significantly altered by the preparation method. So whether we were looking at plasma or serum, and that was also time related, so how long it took to prep, and temperature and things like that. So once that became clear, we went and started to look at components of blood, like all of the platelets, white cells, red cells. And what we found was that there were cytokines in every component, and looking back, we probably shouldn't have been surprised about that. And we developed methods to enable us to look at cytokines in a lot of the components of blood. And our view at the time was that this would be something that people would be quite interested in, and it would add value. And what we saw was, yes, even things like red blood cells had quite high levels of cytokines associated with them. But the reality was, the more we discussed this with different groups, the fewer collaborators we got. And that was really because people were quite interested in it biologically, but they were not interested in it from a cost and difficulty perspective. So they really didn't want to get a lot more data if that meant that it was going to cost a lot more money and take a lot longer. And the biological relevance of it was really kind of unclear. So we spent several years working on projects looking at fractionating whole blood and analyzing cytokines in various sorts of diseases. But essentially what we decided was that we needed to simplify, and we opted for a mixed sample approach. And so what I really mean by that is that we would take venous blood when it was available. So in studies at hospitals where venous blood was collected when patients were, say, in clinic, we would take that, but we would also supplement that with dried blood spot samples and collect samples from people at home. And we've done studies on things like lung cancer where we've taken dried blood spots from people at home, or they self-collect them, and we get venous samples from them when they're in clinic. And we started to look at cytokines from dried blood spots, and the few graphs that you can see here on the right-hand side of this slide kind of indicate what you see. So in a lot of cases, cytokines are present at quite low levels in things like serum or plasma, and can often be present at much higher levels in whole blood that you would get from something like a dried blood spot. There are also ones like ITAC that you can see there on the top left of the graphs, where we were unable to get it to come out of the dried blood spot. So we know it's there because if you look at the cells from venous blood, you can detect it at quite high levels, but it binds and doesn't come back out. And we didn't really understand what was happening at that point. We spent quite a long time looking at stability of cytokines. So we've got quite a lot of data on stability. We published this paper with James Rudge from Neoterics, and what we looked at was both time, so storage right out to 5 months, and temperatures from room temperature, 4 degrees, -20 and -80. And what we found, of course, is that room temperature is fine for quite a short time. Same with things like 4 degrees, but -20 and -80 work quite well. And we, I think, have looked at about 40 cytokines in this paper. So that really takes the history through for us to about 2019 or 2020. And then two things happened that really changed how we used particularly Mitra and dried blood spot samples. So one of those was scaling up and going from individual tubes for extraction into 96-well plate. And Cameron Hill in our lab built a device to enable us to work in 96-well plates and remove the tips of a whole plate worth of Mitra in a single go. And to optimize that method, what he did, or what he wanted to do, was use Mitra that we'd already used. So wash them out and reuse them. And his argument was, well, we don't really need to use brand new Mitra for this because we're just optimizing a method. And interestingly, what happened was if you do that and wash the Mitra and then attempt to use them again, they don't fill properly. It's as though, to use a Western blot kind of analogy, they've been blocked and something stops them from filling properly. So that kind of stymied our approach of being cheap and using old Mitra because they don't fill. And our initial thought here was that there was residual cellular material that was blocking the device. So maybe there was DNA or cell membranes, and that was our working kind of hypothesis at that stage. And we kind of parked that and carried on with method development. And then at a similar time, I had a discussion with Mark Malloy, who is one of our collaborators, who's the professor of bowel cancer research at University of Sydney here. And his question was, did I think we could identify things like CD markers or cell surface proteins from a dried blood spot? And because we thought that membranes were being trapped in Mitra, what I suggested to him was, well, what if we took Mitra and washed it and then trypsin digested it? And this really came from quite a bit of literature that's out there on what they call cell shaving. So a lot of it's done from bacteria, but effectively you use an enzyme like trypsin to strip the surface, and that can be in intact cells or cells that have been lysed. And so when you do that, you can recover the extracellular parts of quite hydrophobic multi-pass transmembrane proteins. And Mark and I had done work like this when we were doing our PhDs together back at Macquarie University. So we decided that this was a approach we should try. And when we did that first, what the Mitra were loaded with, because we were working with plasma in another project, Elizabeth Carston in the lab made buffy coat, so effectively white cells and platelets, and we took that and loaded that with a pipette into Mitra, so the Mitra wouldn't have been red like this picture I've got here. And then we dried it and washed it, and we looked at washing with just simple salt solutions like sodium chloride. In our lab, we tend to use lithium chloride. We find it's better than sodium chloride as a wash. And then the advantage of Mitra, of course, is you can centrifuge it and it doesn't fall apart like filter paper does. So what we did was wash, centrifuge it to empty, and you can see that by the color of the tip that things are changing. Once the tip, we'd washed it, we emptied it and then loaded it with buffer and trypsin. And when we then centrifuged out what we hoped were peptides and took that to the mass spectrometer, what we then found, to our great surprise, was we identified 2,177 proteins, which was more than we thought we would get. And interestingly, what we found was that almost 80% of them were cellular in origin. And I guess looking back at it now, we shouldn't have been surprised because we put in buffy coat, which is cells. A bit over 20% were plasma proteins, and you can see on the graph here how that broke down into the different major protein classes. And we then looked at what there were in terms of organelle proteins. So we were not just seeing cellular and things like cytoskeletal proteins, we were seeing organelle and organelle membrane proteins as well. So we started to then look at, in this data set, what we got in terms of transmembrane proteins. So you can see here there were 257 transmembrane proteins identified. So almost 12% of what we identified in the mass spectrometer were transmembrane proteins. And then when you look at how they break up, whether they're receptors or transporters, that's shown on this particular slide here. And then ultimately looking at CD markers, we got 36 CD markers, so 1.7% of the total, nearly 2,200 were CD markers. So that kind of answered our question that yes, you could do this. And we were, at that point, this was in 2020, and we went on and started to look at this for whole blood. And so when you go and look, and you've been around proteomics for a long time, what you find is, of course, that protein binds to lots and lots of surfaces. So I guess we shouldn't be surprised. We've talked extensively with James and Stuart Cushon at Neoterix about this. It's clear that there are binding sites inside devices like Mitra, but the same things exist on things like PVDF membrane. We've done this kind of work with filter paper, and you get similar results. PVDF's not as good. It binds very tightly, but you can digest from its surface. Filter paper works, but it's not robust. So when you go through several washing steps, what you find is, of course, the filter paper starts to disintegrate and fall apart. So we like Mitra because it's so robust and we can go through this kind of sequential process. And when you then start to do that, how we really see it, I guess, is the washing at the start removes what you would call unbound, and then you're left with the bound. So the graphs here show metabolite. So obviously what comes out in that initial wash are not just proteins, but metabolites. So we're currently optimizing these graphs here. If you look on the far right, plasma, and then 4 different dried blood spots that were extracted in different ways. And you can see basically there's not an awful lot of difference between the plasma level of the metabolite and what comes out of Mitra, irrespective of the type of extraction we've done here. So we're currently optimizing this, and what you can then do is go back and use exactly the same tip and trypsin digest and look at proteins from the same tip. So the method kind of summary that we've got here is shown in 96-well plate format. It works just as well if you do it in individual tubes. So we typically would do a small volume extraction from a 30 microliter Mitra. We would do a 90 microliter extraction in PBS. Initially, we would have used PBS tween, but tween's not really great for mass spectrometry, and there appears to be little difference in recovery of things like cytokines or metabolites, whether you have a detergent there or not. So that initial extraction, we would do things like immunoassay or store it away in case we want to go back to it later. In terms of the washing, we've looked at pretty wide range of things that you can wash with. So everything from salt solutions like sodium or lithium chloride, detergents, things like urea, thiourea, organic solvent, and they all take a different fraction and are more or less stringent. We tend to use lithium chloride because it's very easy to do and it's very consistent, and we get highly reproducible results doing it. We then do protein digestion in the tip, and we use solid phase extraction for cleanup. And the mass spectrometry that we're doing is usually DIA mass spec on a Thermo Fisher Orbitrap HFX. And we use about a 1 hour or 70-minute gradient most of the time. We've done work on other instruments looking at shorter gradients, and of course that works, but you get fewer identifications. And then we started to look at how this would work for things like plasma and serum and other samples. We've done quite a lot of work. If people are interested in any of this, if any of you want to know more, you can always contact me, and I'm happy to discuss the kind of things that we've done. What you're seeing here on the left is single spun plasma versus whole blood. And the highest bar is where that single spun or whole blood has gone into Mitra and been dried, washed. So using our optimized method. And the small ones are where we've either done a liquid digest, so it's never gone into Mitra, or it has gone into Mitra but has not been washed. And you can see that what you'd see there is far fewer identifications, both in whole blood and in single spun plasma. And in single spun plasma, you see a lot of the extracellular vesicle material that is there in single spun plasma. So that's probably a little bit hard to read maybe, but membrane bound vesicles and extracellular vesicles are quite highly represented in that single spun plasma from the optimized method, where you've dried that plasma into Mitra and then washed it and digested it. And the same is true from whole blood. And then on the right, you can see an additional sample, which is double spun plasma, which is the blue line, and you get about 1,500 IDs. The yellow line here is single spun plasma, and then whole blood, where we get almost 4,000 IDs. And then the CVs, we typically get CVs of less than 15%, often less than 10%. But double spun plasma, as you can see, is not as good. So in fact, where there's cellular material in our hands with devices like Mitra, we get quite good results. It works for things like double spun plasma, but it's not as good probably there as some of the bead-based depletion methods, and we've got a bit of data on that. The other thing that we became interested in then was, clearly these devices work as sample prep tools as well as sample collection tools. So would they work from things like frozen blood? And they do. And this paper we published fairly recently on non-small cell lung cancer. We had about 18 samples of lung cancer blood and a same number of age and sex match controls. So this was done with the frozen cell pellet after plasma had been prepared, and that cell pellet was just frozen. The residual plasma wasn't removed, the buffy coat wasn't. So you've basically got, it's effectively like cell concentrated whole blood that's been frozen. And it's quite straightforward to thaw that kind of sample, load it into Mitra, dry it, wash it. So in this case, we got about 4,000 IDs for each sample, and we identified about 500 proteins that were different. So that's, like I say, we published that recently. So what we've really evolved to is our kind of platform for biomarker discovery. And effectively what we do is we like to work with a large-scale screening program. Our focus is really around early stage cancer diagnostics. And of course, what I'll tell you about today is some of the stuff we're doing in colorectal polyps. So basically, if you can get an at-home blood sample from a patient when they're going through a screening program, and you can get the clinical data that tells you whether the person either has cancer or, in the case of things like polyps, pre-cancer, then hopefully what you can do is go through and find biomarkers from the blood that correlate with that. So that's what we're doing. The reason that we're looking at colorectal is really that's one of the major projects that our collaborator Mark Malloy works on. So we got involved in this quite a few years ago. And colorectal cancer is the second leading cancer killer. It kills about 1 million people in the world each year. And a couple of million people are diagnosed with it. And it's increasing. So despite the fact that there are screening programs, the prediction is that there'll be 1.6 million deaths a year by 2040. However, if you can detect it early, the survival rate is quite high. And in fact, if you can detect polyps, then you can effectively 100%, almost 100% prevent cancer if you can detect polyps before they've become cancerous. The difficulty is the type of stool sample testing that's sent out in government screening programs in quite a few countries is not particularly effective at detecting polyps. It's quite good at detecting cancer, and also the compliance rate's quite low. Most countries struggle to get more than 30 or 40% of people to do it, really, because most people don't want to poke a stick in their own poo at home and then put that in a tube and send it away. So that's the kind of thing that's holding back, because really we could completely eradicate colorectal cancer if enough people were to have colonoscopies, but something has to drive them to take a colonoscopy. And the reason colonoscopies are so useful is that if you've got a polyp, so for example, on this diagram here, if you've got something that's either at that 0, 1, 2, or even 3, it can be removed in a colonoscopy. But once you've got invasive, it's invading through the mucosa of the colon, you can't really remove it in a colonoscopy. And they're the things that are detected well by current stool and blood tests. Excuse me. So the tests are not really great at detecting polyps. So that's why we're looking, and I'm sure other groups are looking at detecting colorectal polyps, because that's the thing that it's so much cheaper. Here in Australia, a colonoscopy costs a couple of $1,000, whereas surgery for even early stage cancer is far more, 20 times more expensive than that. I won't read that. You can have a look at that later. The two guys here, Dan and David, are gastroenterologists who we're collaborating with, and Mark Malloy is there on the right. So the original study that is still running at Royal North Shore Hospital, where we are in Sydney, Mark is one of the principal investigators of that, and that study takes venous blood samples on the day of colonoscopy. And what we do, they're processed into plasma and cell pellet, but we also take a whole blood sample and load that into Mitra. So we've used Mitra in this scenario on that study as a sample prep tool. We've looked at a range of cytokines, and there are certainly cytokines that do not show a difference or a very low level in plasma, but from dried blood spots, not only are they detectable, but there's a significant difference between polyp and no polyp. And then in terms of mass spectrometry, where we got to the last time we ran a batch of samples from that study, we were identifying about 2,600 proteins per sample. We'd identified a few hundred that were different, and about 19 of them had large fold changes. And interestingly, the pathways that those proteins are in are cancer-associated pathways, even though these are not classified as cancer. And then that study as well, what Mark is looking at, we didn't do this work, is the actual mucosal microbiome. So they're getting not just blood from these people, they're getting samples, biopsies of their colon, the polyps, and healthy tissue. And what Mark's looked at is the microbiome in that mucosa. So this paper came out not that long ago and showed that there were bacteria that were associated with polyps, things like Ruminococcus narvis. So the study sounds okay, but there are several drawbacks with that study. One is it's venous blood, and really venous blood, of course, is not an at-home sample that people can routinely take. It's expensive, logistically difficult to do, hard to get longitudinally, all the things that this group would understand. The other thing that I think is interesting, although we haven't really looked at this, is, of course, when the people come in for their colonoscopy, the bowel prep has already been done. So they've drunk that solution that causes them to basically empty out their colon and their bowel. So my thought would be that that's, at least for some of them, likely to change certain things in their blood. So maybe that's not a great screening tool either. But the really major issue with the study is there's a very low recruitment rate. So we got our first sample in that study in August 2020, and we've only had about 44 samples a year. And that's nowhere near how many colonoscopies are done at the hospital. The reason is that people are coming in, they really probably don't want to have a colonoscopy, and then they get asked if they'll be in a study and can they give a blood sample, and a lot of them just go, yeah, I'm not interested, I just want to have this done and leave, because they've got to fill forms in and they're just not into it. And that's not unique to this study. We see that kind of thing in lots of studies. So to get to significant numbers, what the power analyses said that we would need was hundreds more samples at the very least. So what we designed was a study that was entirely at home, where we would get people before they went into their colonoscopy, 2 or 3 weeks before, so they were eating normally, they hadn't started their bowel prep. So that study we initially envisaged as a 1,000 person study, but it's grown since then. And we asked the people in that study as well to give us a dried blood spot, also to give us another stool sample. And the stool sample is not to look at hemoglobin in the stool, it's to do gut microbiome analysis. And the initial work that I'll show you a snippet of here was done by Microba, who are in Brisbane here in Australia, and they do metagenomic microbiome analysis. And the blood samples were all taken on the at-home sampling kit that's got 2 30 microliter Mitra in it, and were analysed. Like I say, we haven't yet looked at cytokines. Everything we've done so far is LCMS on the Thermo Orbitrap. And the group that Dan and David that I showed you before, owners of and work in, is called the Colonoscopy Clinic, and they're based in Brisbane. They do about 10,000 colonoscopies a year, and many of the people that they do them on are part of colonoscopy screening programs. So they've had polyps previously and they come back every few years for another colonoscopy. So they have been great because they were able to go out to their entire kind of patient list who were scheduled for colonoscopies and ask them to come into the study. So completely different to the hospital study, we started recruitment with the Colonoscopy Clinic in February last year, and by December last year we had recruited 1,400 people. So they wrote to about 3,000 or 4,000 people. Nearly half of them said yes. And this funnel plot here shows, so we've got 1,900 people were screened, 1,464 enrolled, and so on and so forth. So we ship the kits out. Most of the people are around the Brisbane area, and Elizabeth Carston, who is our principal scientist, lives in Brisbane, so she's done an awesome job of coordinating this study, and the samples get shipped back to her. And then we ultimately get the colonoscopy report and the biopsy report, and what the team are then doing is putting together quite a large database of all of the clinical data, not just from this colonoscopy, but previous ones. And we have a lot of detail on all of these people, much more than you would get in a conventional hospital system. In terms of the microbiome, this is just from the first 200, so 100 people in the polyp group and 100 people in the control group, and I'll show you how those groups break up. This is tremendously complex. I'm only going to show you this little snippet of it here. And what you're really seeing here is what's called richness, or the Shannon index of diversity. So that's exactly what it sounds like. How many microbial units are there, different species, or operational taxonomic units is the fancy phrase for that. And interestingly, the trend towards higher diversity is not necessarily a good thing. So previous studies have suggested that the microbiome of colorectal cancer patients has higher diversity than controls. And you can see that trend is present here, both in richness, this graph in the middle, and in the Shannon diversity, which is the one on the right. And that's comparing people with polyps that are between 5 and 9 millimeters and polyps that are 10 millimeters and larger. So what you're seeing is the larger the polyp becomes, the more diversity there appears to be in their microbiome. And there's quite a bit of literature on the role of different bacteria in creating inflammatory niches and being involved in the genesis of polyps and colorectal cancer. So we've got samples, of course, from basically 1,400 people. So there's a lot more microbiome work to do, and we're looking at whether we'll do that with metagenomics or long read 16S, which may be the way we go initially. So in terms of Mitra and at-home blood sampling, out of that 1,400 people, we've got 1,159 samples are usable, so almost 86%. And the table here shows that. So people who filled both completely was almost 70% of people, 932. People who filled one and then got a partial fill on the other one was another 16%, and one full but kind of no attempt on the other one was only 7 people. So we were pretty happy with that, and we went with 30 microliter Mitra, knowing that, you know, some people struggle to fill both, and maybe in future studies we'll use smaller samplers, because really 30 microliters is an awful lot, and we don't really, you know, we're loading quite a small amount of material into the mass spectrometer. In terms of how that breaks up, what you see, I've got notes on here. So this is the graph on the left of the grey is female, the red is male. So you can see there's not very many people with no polyps. The majority of people in the study have polyps. And we were surprised about that up until maybe a couple of years ago. But the critical thing to understand is that a lot of these people are known to have polyps. They've had them before, so it's not unexpected they'll have them again. And the other thing is, not all colonoscopies are the same. So the withdrawal time of the scope correlates with the number of polyps found. And the average withdrawal time in the colonoscopy clinic cohort, which is the vast majority of what we've got, is 19 minutes, almost 20 minutes. Whereas the government recommendation here in Australia is that a colonoscopy should take at least 6 minutes. However, you know, most gastroenterologists would take considerably longer than that. And the studies I've looked at, most of them done even in public hospitals, are over 10 minutes. So the more you do, the longer you take, the more polyps you find. However, 10 millimeter polyps and larger is what's considered the clinically at-risk group. So that's what they're really looking for, particularly in the public hospital system, because they're trying to get through as many as they can. What we found in the private system is you tend to see a lot more data on smaller polyps. And so the groups break up, as you can see here. So 132 people with no polyps. The largest group of people with polyps up to 5 millimeters. And so we've called that the control group, because those small polyps would be considered not risky by the kind of clinical standard that's applied in most of the developed world. And then what we've called the polyp group is people with 5 to 9 millimeter or 10 millimeter and above polyps. And then if we go to the next slide here. So like I've said, this is the reason that people had a colonoscopy, and half of them are having it because they've had a polyp previously, another 20% because they've got a family history, 20% of them come in because they've had some kind of gastrointestinal symptom change, and so they're having a colonoscopy for that. The fecal occult blood or the immunoassay for hemoglobin kind of tests account for less than 10% of the people in this particular study. So ultimately we will need to look at a larger group of those people, because some of those people will be coming in for their first colonoscopy. So we need to look at increasing that group. And then what you see on the right is the sizes and the types. So tubular adenomas, serrated sessile lesions, hyperplastic polyps, or other sorts are here. So tubular adenomas is the biggest polyp type that we see, that and serrated sessile lesions. So the next one, I won't talk about this at all. People can read this if you're interested in it. So this is the type of data analysis that we do after we've got the mass spectrometry data, how we evaluate the data and what we're looking for. So in terms of this larger study, we're running at the moment, we run in blocks of 200. So the 200 we're running right now will take us up to 600 analysed. So what you're seeing here is data from the first 400 that we've run out of the 1400. And unfortunately, I can't really disclose any of the actual proteins that we're seeing, because we've not filed a patent on that yet. But if anybody is particularly interested in this polyp study, if you contact me and there's an opportunity to collaborate or discuss, we could potentially do some deeper discussions under a confidentiality agreement. Happy to talk with people about that. What we're looking at when we do the differential expression analysis is both protein and peptide level, because of course from the mass spectrometer you're seeing peptides, and so often you're only seeing fragments of the protein. It's uncommon to get coverage of the entire protein. So we find that in our hands, the peptides produce better results doing this kind of differential analysis than protein level differential expression. And of course, on this volcano plot, what you're seeing is the differential peptides are in the red boxes here, whether they're decreased or increased. And this is one of the ROC curves that we've got. And we've seen similar results even in the hospital study and in the initial analyses we've done. We typically get sensitivity around about 90% to distinguish polyp, no polyp. In this case, what we're looking at is the control group versus people with 10 millimeter polyps or larger. What you see here is the specificity is in the high 70s. And at the moment, that's the thing that I think we need to focus on. We have not yet got to a point where we've got very high sensitivity and specificity with a panel, and I think that will come hopefully once we've analysed more of the samples and we can discriminate more in terms of the panel that we're looking at. What we do see, and there's more detail here, it's similar to what we saw in the hospital study, is that we hit a lot of cancer-related pathways in the differentially expressed proteins, and I've listed out the primary cancer-related pathways that we see here. So when you go and look at the individual proteins in there, they're very interesting. I find it quite encouraging that we're seeing cancer-related pathways and proteins that are known to be involved in the development of cancer. So in that sense, it's quite an interesting validation, at least in my mind. The other interesting thing about whole blood that I think is an advantage is, like I showed you in the first analysis that we did of the Buffy coat, in this case, 60% of the proteins that we identify are not in plasma. So the Polyp one here is a differential expression set of about 300 proteins that we've got from one of our analyses. The lung cancer one is the 500 odd proteins we saw in our lung cancer study. And the O-Link circle is, what I've done here is just downloaded the O-Link Explore panel and compared. And what you see is that there is overlap, but 60% of the differential expression that we're seeing is not in the O-Link panel. And it's a very large panel. It's about 5,500 proteins. So I find that quite interesting. This method is producing things that are unique because we're dealing with cellular samples, effectively whole blood. What we've looked at from the most recent data set is what panel size works in terms of how many proteins, because to commercialize something like this, you're not going to look at 300 proteins. You need to bring it down to preferably less than 20, and even if you can, less than 10. So we've looked at, in this case, 3, 5, 10, 15, and 20. 10, at least according to the criteria we've used, produced the best panel. So in this case, its sensitivity was 84 nearly, specificity 76. So there's variation depending on what question you ask and what panel you use. That's really the point here. You can see that with other panels, we have better specificity, but lower sensitivity. So it really depends what you're going for. And I think our goal, certainly in my mind, is to get both the sensitivity and specificity as close to 90 as we can. Certainly in the commercially available tests, if you look on the next slide, we have very good sensitivity compared to the other tests that are out there. So the blood and the stool test, the FIT test, Colorectal, which is Exact Sciences in the United States, their polyp detection is the best on the market at the moment, but it's about 43%. We're double that, but and there's variation. We see different panels, different markers depending on male or female, and the particular subgroups that we look at. So the goal really, I think, for me is to now take this and look at getting our sensitivity higher as we get more and more samples analysed out of that 1,400. And then we will certainly look at bringing in cytokine markers and metabolites as well. So the next steps in this particular study for us will probably go, I think, to long read 16S sequencing for microbiome analysis, because that's a lot cheaper than metagenomics, and we can do probably the whole 1,400 samples there. We'll bring in metabolomics. We've basically got a method worked out now where we can do metabolomics and then proteomics from the same tip. We'll continue with the mass spectrometry and also bring in cytokine analysis. The target, like I said, is 10 millimeter polyps. We'd like a single panel that predicts 10 millimeter polyps with good sensitivity and specificity. And it's likely there'll also be distinct panels for men and women, which may be useful for particular age groups, for example. So that kind of thing, I suspect, will happen as well. And then to summarize where we're at, I think for us, the use of Mitra has been fantastic. It's really enabled this study to work. There's little value having a group like the colonoscopy clinic that can reach so many patients if you can't get samples from them. So this has worked incredibly well for us. We've had very high patient compliance. We've got an agreement with them to go back and do additional recruitment if we need further groups for validation. It's very low cost compared to bringing people into a hospital and getting a venous sample. Hopefully what I've shown you in enough detail is the same device can be used for sampling and sample preparation. We get very deep coverage compared to plasma. And like I said, more than half of the markers we see are novel. They're not found in plasma. So that's the end of my presentation, and I'll hand back to Phil.
Yes, thanks, Ben. So we have a time for a few questions, so please do continue to log your questions using the chat function if you have any for Ben. I'll just whiz through the ones that are in there at the moment. So Ben, here's one that's come in, which is what is the buffer that you use for your cytokine analysis?
To extract, we just extract in PBS. So that we used to extract in PBS tween, but what you can do if you don't want to go on and do mass spec, but the tween interferes and is hard to wash away. So we tend to just use PBS for the extraction, and we do most of our cytokine work on Luminex. We've got access to a Biorad Bioplex instrument, and we tend to use a millipore, things like their high sensitivity T cell kit we use. We've also got an Olink Q100 machine, but we don't use that as much. Yeah.
Okay, thank you. And how are you collecting, or how is one collecting the at-home stool samples?
Oh, good question. The Microba, we use their collection kit, which is a small tube, very similar to one of the kind of government screening kits. So basically you use a piece of toilet paper with some stool on it. There's a little stick inside. You undo the tube. There's a little stick with a little brush on the end of it. You brush a small amount of stool onto the stick, put it back in the tube. It's got preservative in it, and that will keep that sample stable at room temperature for quite a long time, and they're posted back. So we send out a post pack with the at-home sampling. So people do the blood sample and the stool sample, and they both come back through the post. And the part of why we did the initial 200 instead of going to a much larger group was to check sample quality and make sure what we were getting was good. But it was, Microba have a lot of experience with sampling. So yeah, those kits are commercially available from several different suppliers.
Okay. And here's sort of a comment, really, that would be good to get your input on. So back on slide 30, which showed the microbiome results, let's pull that up. So there seems to be a large overlap between the 5 to 9 compared to the 10 sized polyp. Can you comment on that?
Yeah, I mean, there's definitely overlap. I mean, it's statistically significant, but when you think about it, those two groups have both got polyps. It's really just the size that is different. And what we were interested in was that there was a trend towards higher diversity in larger, more clinically risky polyps, which mirrors what's been seen in the literature. I think the other thing to bear in mind here is this is only 200 samples. So that that set of data that you're seeing here is really from 100 people. There was another 100 who were in the control group. So this is a small data set. It's indicative that there might be a trend. Once we analyze a lot more of the 1400, we'll have a much better sense of what is that trend really like, if it's really there in the whole group, and which bacteria are the ones contributing to the increased diversity, if it's really true. But I think it will be. The literature is reasonably clear that there is increased diversity in colorectal cancer. So it may well be that that's happening at the polyp stage. So really, this was an indication that something's happening. That's not meant to be definitive.
Okay. Thank you. And what do you think of the potential of dried urine spot as a second tool?
We've talked with James at Neoterics about this. We've never tried it. I think given what we understand now about how devices like Mitra work, it would work in a very similar way. You would get material that bound quite tightly to the device. You'd be able to wash away the salt and abundant proteins in the urine and do exactly what we're doing. I've worked with urine in the past, and it's quite low protein concentration, of course, compared to blood. So whether a device like a Mitra will give you sufficient material, I don't know. But in principle, it would work well. And it's certainly non-invasive.
Okay. And have you also tested Whatman 903 for protein work?
Yeah, that was the filter paper we tested, I think, in the initial stages when we were looking at this. We published a paper with Mark early in the development of this, and I think there's some data in that, and I think that's the filter paper we used. It does work. You get protein binding to it. You can wash it and get high levels of protein out of it once you've digested it, but you've got to be a lot more gentle with it because, of course, it starts to fall apart. So you can't be as aggressive as you can with something like Mitra.
Okay, well, thanks for that. And I think, yeah, one final question. What's your plan for validation cohorts in the polyp study?
So what we're doing at the moment is looking at, of course, these blocks of 200 out of the 1400. Our goal really is to look in commercialization in the United States. So we're almost certainly going to have to do another study with a validation cohort in the United States. So we're talking to groups over there with a view to how that will be done. We've got an agreement with the colonoscopy clinic here in Brisbane that we can recruit more. So if we want to do a validation group outside the 1400 we've done, we can certainly recruit more people there. So it really depends. We've clearly got to do that, but we also need to finalize the panel or panels that we want to look at first. But I expect that will start into some validation either later this year or early next year. So we're exploring that now.
Excellent. Oh, yeah, thanks, Ben. Thanks for a great presentation, great webinar. And thank you for everyone for attending today. If you're not already, please do follow the PCSIG on LinkedIn. There's a QR code here on the bottom right. And you can also watch all of the past webinars that we've done on the PCSIG website. So please take a look there. And thank you again to our sponsors for making the webinars possible. Next webinar will be in July, and we'll be publishing more information on that shortly. So just need to say thanks again to you, Ben, for a great webinar. And thanks again, everyone, for watching. And yes, until next time. Thanks. Bye.
Thank you.